Hello again. We're going to continue our explanation on oxidative phosphorylation. Now, we have just talked about, in part number 2, we talked about the transfer of electrons from one electron carrier to another, and it's worth saying that when electrons are transferred from one electron carrier to another, what actually happens is the release of energy. So upon transfer of electrons, if this is an electron, and it be moved to the second electron carrier, and the second electron carrier will move it to the third electron carrier. As we said, that electrons are being transferred at gradually decreasing energy levels. So there is a gradual decrease in energy level, meaning that every electron carrier will receive the electrons at a lower energy level. And if this occurs, then there will be release of energy. What are we going to do with that released energy? That released energy will be utilized by the electron carrier itself to pump protons against concentration gradient. And that's what you see here, that every electron carrier, after having transferred the electron to the next electron carrier and the release of energy, will pump protons to the outer mitochondrial space, against concentration gradient. So the process of transfer of electrons releases energy and is considered an exergonic process, whereas the process of pumping the protons is an endergonic process because it utilizes energy. And the same is repeated along the electron transfer chain. So by the end of the electron transfer chain, we will see that we have accumulated the protons against concentration gradient in the outer mitochondrial space, and that there is a lot of stored potential energy in those electrons that have accumulated here. Of course, the presence of that selectively permeable membrane will help in the creation or preservation actually of that concentration gradient of protons. I'll open a screen now and explain something or recapitulate what we said in a very specific way, because this is an important part that needs recapitulation. When we talk about the electron transfer chain, there are three types of coupling. So here we would summarize and say coupling processes in electron transfer chain, or coupling processes along the electron transfer chain. So number one, we have seen coupling oxidation with reduction, coupling oxidation to reduction, because every time something is oxidized or a molecule is oxidized, another molecule will be reduced. Parallel to this, the three coupling processes occur as the electron transport chain is proceeding, meaning that the three coupling processes occur hand in hand. They are parallel to each other. So the second coupling process is coupling of endergonic processes with exergonic processes. Coupling, exergonic first, of course, coupling of exergonic processes. For example, the exergonic process here was the electron transfer, to endergonic processes, processes, and the prototype of those endergonic processes is proton pumping to the outer mitochondrial space. Proton pumping to the outer mitochondrial space, to the outer mitochondrial space. You might ask me, or you might be wondering, why are we saying that when there is electron transfer and release of energy, which is an exergonic process, why do we say that the endergonic processes are only the process of proton pumping? This is very true. You're right, because the endergonic processes are not only proton pumping to the outer mitochondrial space, but this is the most important one, or actually the more important one. But there is another one that is also important. I mean, this is not the more important one, but the more famous to say. But the other one is as well important, because the other endergonic process is the reduction of the next carrier, because the reduction or gain of electrons by the next carrier, gain of electrons by the next carrier is also an endergonic process. So when we say that we have coupling of oxidation with reduction, this is coupling of exergonic process with endergonic process. So the third coupling is, now when all this energy is released and when all the protons have been pumped, the last stage of the electron transfer chain that occurs along the ATP synthase enzyme, the ATP synthase enzyme. Is it coupling? Yes, it is coupling. Why? Because the main function of the ATP synthase enzyme is to synthesize ATP. How does it function? Now, this is the channel that is called ATP synthase, and we will see it again, but I can't move back to the previous slide because the screen would disappear. Now that you have pumped all the protons in the outer mitochondrial space, this is the inner mitochondrial membrane, and you have accumulated protons in the outer mitochondrial space. When enough protons have been accumulated outside the inner mitochondrial membrane, or actually between the inner mitochondrial membrane and outer mitochondrial membrane, you now have what we call the electrochemical gradient. Why is it called an electrochemical gradient? Gradient means difference, meaning that now we have, and this is the inner mitochondrial membrane, by the way, that is very similar to the cell membrane. Now, is there an electric gradient? Yes, because we have a lot of positive charge outside and we don't have similar positive charge inside, or maybe we have fewer positive charge inside. Is it a chemical gradient? Yes, when we say chemical gradient, we mean a difference in concentration. Yes, it is also a chemical gradient because we have a concentration gradient actually. So when we say chemical gradient, we mean, when we say chemical gradient, we mean concentration gradient. Yes, there is a concentration gradient because there is a difference in concentration between the hydrogen ions outside and the hydrogen ions inside the matrix. This electrochemical gradient will allow the protons to pass down the ATP synthase down concentration gradient in a process that is called chemiosmosis. Chemiosmosis. So it looks like osmosis in the fact that it is a passive process from higher concentration gradient to lower concentration gradient, and that it does not utilize any energy, but it is a very specific type of diffusion because it is chemi-diffusion. It is diffusion of charged ions like the protons. Okay, so what we actually did is that we kept on pumping the protons outside using the electron carriers, and then when they accumulated outside, we allowed them to pass through the ATP synthase when a sufficient electrochemical gradient had been formed. What happens when they pass down through the process of chemiosmosis? They release their energy. They relax and release their energy, and that energy will be stored inside an ATP molecule by combining with ADP to form ATP. So what kind of coupling do we have in the function of ATP synthase? The coupling we have for the function of ATP, we have actually two types of coupling for ATP synthase. Again, we couple an endergonic process with an exergonic process, or actually, we have to start by exergonic. So we couple an exergonic process. What was the exergonic process in this step of the ATP synthase? It is the release of energy in chemiosmosis. It is the release of the stored potential energy, which is chemical energy during which phase? During chemiosmosis. It is the release of potential energy during chemiosmosis, and we coupled this exergonic process with the endergonic process, and the endergonic process was the formation of ATP. Is that the only type of coupling that is done by the ATP synthase? No, there is another type of coupling done by the ATP synthase, and it is the coupling of oxidation to phosphorylation. Actually, the coupling of oxidation to phosphorylation is not a function of ATP synthase alone, but it occurs along the whole length of the electron transport chain. But why do we say this here? Because ATP synthase is the stage at which the phosphorylation occurs, which is the addition of a phosphate group to ADP. So here, actually, it's not only energy, plus ADP, but it's also energy plus phosphate plus ATP. So this is very important to know, that we have coupling of oxidation to reduction, we have coupling of exergonic processes to endergonic processes, and we have coupling of oxidation to phosphorylation. And now we have, we will open, or we actually will erase everything on the screen and say something very important, which is, actually, I have to reopen the screen. We will say something very important. When we said that here we have the inner mitochondrial membrane, and here we have the outer mitochondrial space, and that the electron carriers on the inner mitochondrial membrane use the energy released from the flow of electrons to pump protons, then the electron, the function of the electron carriers is that they are, number one, they are protein enzymes. They are enzymes, and therefore they are proteins. Number two, they are couplers. They are couplers because they couple oxidation to reduction. And number three, they are proton pumpers. Okay. Here, couplers, or coupling oxidation to reduction, is because they are electron carriers. Okay. Now, when we pumped the protons outside, we are accumulating them outside, and we are doing this on purpose. We are accumulating them against concentration gradient using energy derived from the transfer of electrons, and we have created an electrochemical an electrochemical gradient. And here there is a very important concept that you have to know, which is that this electrochemical chemical gradient would not have been created and preserved, would not have been created and preserved if there weren't a plasma membrane. So here, the presence of a selectively permeable membrane, the presence of, this is a very important concept to understand, the presence of a selectively permeable, the presence of a selectively permeable membrane creates the gradient and creates the electrochemical gradient. And then after creating the electrochemical gradient, preserves the gradient. So it does both. How does it create the gradient? Through the proteins that are present, through the cell membrane proteins. So if you didn't have those electron carriers, you couldn't, if you didn't have those electron carriers, you couldn't have created that selectively permeable membrane. And meanwhile, it preserves the gradient. It preserves the gradient because it prevents simple diffusion of hydrogen ions. And it says to the hydrogen ions, simple diffusion of hydrogen ions. And it says to the hydrogen ions, if you want to go back into the matrix to release your stored energy and help the cell, the mitochondrion to form ATP, then just pass through the ATP synthase, okay? This is an important thing to know. The other important thing to know is that you sometimes have a wrap-up question like, for example, proteins of the cell membrane, proteins of the cell membrane or cell membrane proteins. So don't forget to add this previous piece of information to your talk about the selective permeability of the membrane. So here, proteins of the cell membrane with enzymatic action. How many proteins of the cell membrane have you studied up till now that have an enzymatic action? You can, of course, like pause the video and start reflecting on this and then say the answer. So proteins of the cell membrane that have enzymatic action, we have seen, for example, adenylate cyclase in the G protein coupled receptor pathway. We have seen tyrosine kinases. We have seen tyrosine kinases, okay? We have seen the complexes of the electron transfer chain, the electron transfer chain complex proteins. They are enzymes from number 1 to number 4. And finally, we have the last member of the electron transport chain, which is the last member of the electron transport chain, which is the ATP synthase enzyme. The ATP synthase enzyme, of course, as you know, has an enzymatic function. The ATP synthase is not only an enzyme, but it is a channel for the hydrogen ions, and it is a coupler because it couples oxidation with phosphorylation, and it couples endergonic with exergonic processes. What's the final stage in electron transfer chain now that we have transferred the electrons? So here, what are the events? Of course, the electron transport chain is a biochemical pathway. And as we said, that we are going to recapitulate. It is a biochemical pathway. So it is a series of biochemical reactions. It is a series of biochemical reactions. And the most important thing in that series of biochemical reaction is that energy is released in steps. Energy is released in steps, and this applies to all biochemical pathways that include exergonic reactions, splitting the process into A series of chemical reactions helps the energy to be released in steps, and therefore, when the energy is released in steps, the energy can thus be stored. So the electron transfer chain, as we said, leads to a more efficient utilization of the reduced energy. A more efficient utilization of energy. A more efficient utilization of energy. You don't lose energy and you store it in ATP. What's the final electron acceptor? Why do we say that? Why doesn't the electron transfer chain work in the absence of oxygen? Because oxygen is the final electron acceptor of the electrons that are transferred in the electron transfer chain. So the answer is that oxygen is the final electron acceptor in the electron transfer chain. Oxygen is the final electron acceptor in the electron transfer chain. And if there is no oxygen, this means that if there is no oxygen, what would happen? Electron carriers, and therefore, if you go backward, if you go retrograde, reduced NAD and reduced fat will not unload. They will not unload. They will not get rid of their hydrogen and electrons, so they will stay reduced. So Krebs cycle would stop. And by the way, I'd like to highlight something I said at the end of part 1 of cellular respiration, which is how many electrons and how many protons are carried by NAD. As I told you, the chemical formula of NAD is NAD+, and this is because it has a missing electron. So when it gains 2 electrons from the carbon skeleton of glucose, actually we can write it like this. We can translate represent every hydrogen atom as a proton in the nucleus of the hydrogen atom and the electron outside. So this is the first hydrogen atom and this is the second hydrogen atom. One of these electrons will be used to reduce NAD. So this electron will reduce NAD. So NAD will be converted into a neutral molecule. But by doing what? By gaining an electron. So what will remain will be a hole, after having utilized that electron to neutralize the charge. What remains actually of the 2 hydrogen atoms is a whole hydrogen atom and a hydrogen atom without a proton. This one can be written H because it is a whole hydrogen atom, and that one can be carried H+. So actually it is H and H+. So actually what is carried by the molecule of NAD here, if you calculate the electrons and protons that are carried by NAD, you will see that NAD has carried 2, we say it like this. Let me see how they wrote it on the slide. Yes, it carried 1 proton and 2 electrons. It carried 1 proton and 2 electrons. Why 2 electrons? Because NAD has received 1 electron to neutralize its charge, and then has received another electron in the whole hydrogen atom. So this can be written NAD. Okay, here we have neutralization, and then here this is a whole hydrogen atom. So let me use another color. red color, for example. So here you have a whole hydrogen atom that is written like this, okay? And then here we have the electron that neutralized the already present positive charge. And what remains is a proton. So according to this simulation, we say that it received first electron, second electron, and a proton. Okay, 2 electrons and 1 proton. Now, let's read the slides in order to see whether we missed anything on the electron transport chain. So again, the electron transport chain is a series of enzyme proteins as well as coenzymes. It's present on the inner mitochondrial membrane. They are electron carriers and they are proton pumpers. It couples oxidation with reduction or exergonic processes with endergonic processes. And the energy that is released from the exergonic flow of electrons, so the flow of electrons is exergonic, is used to pump protons against concentration gradient from where to where? From the matrix to the outer mitochondrial space. Thus, we have established a proton gradient, and this energy will be stored in the pumped protons. So the pumped protons store potential chemical energy, and this will be released when they pass passively down concentration gradient by facilitated diffusion through the ATP synthase, and this energy will be stored in ATP molecules. So the process of diffusion of the protons down the ATP synthase is called chemiosmosis, as I said. It is important to know that the electron transport chain does not make the ATP directly, like in case of substrate, direct substrate level phosphorylation, but it helps, it prepares for the occurrence of ATP formation through ATP synthase. So the direct enzyme, this is very important to know. So we have several steps of formation of ATP. So here, if we talk about ATP formation, on this screen, we're going to discuss or to summarize the ATP formation during cellular respiration. So it is ATP formation, okay, during cellular respiration. We have direct substrate phosphorylation and oxidative phosphorylation. Or actually, let's first say that ATP formation during cellular respiration can be divided into direct substrate phosphorylation, which is an enzymatic direct substrate level. It has several adjectives before saying phosphorylation. So you have direct, substrate level, enzymatic, because it requires enzymes, enzymatic phosphorylation. And we have indirect phosphorylation through oxidation. Indirect phosphorylation through indirect phosphorylation, okay, through oxidation, through oxidation. So the indirect phosphorylation through oxidation is the formation of ATP when reduced NAD and reduced fat deliver their electrons along the electron transfer chain until ATP is produced at the end of the day. So this whole process of losing electrons from reduced NAD and fat to form ATP at the end of the electron transfer chain is the indirect phosphorylation. Why do we say that it is indirect? Because this process of transfer is not direct addition of phosphate to the substrate. And why do we say that it is oxidative? Because there is loss of electrons, which is oxidation. Whereas here, direct substrate level enzymatic phosphorylation, here you add a phosphate, you directly add a phosphate to a substrate. Along cellular respiration, we have seen direct phosphorylation twice via two types of enzymes. The first one are the kinase enzymes that we have seen in glycolysis, and we have seen it once per turn of Krebs cycle. And the other one is the function, the last step of the electron transfer chain, the last step of the electron transfer chain, which is the function of the ATP synthase enzyme. So it's very important to understand that although ATP synthase is the last step of the whole process of oxidative phosphorylation, yet ATP synthase per se, ATP synthase on its own does the reaction of direct substrate phosphorylation, because when the protons pass through the ATP synthase, and actually it's interesting to know that when the protons pass down the electron, down ATP synthase, it rotates, and that rotation helps in the process of direct phosphorylation. So you're actually adding phosphate directly to ADP plus the produced energy to form ATP. So again, although ATP synthase is the last step of the indirect process of oxidative phosphorylation, but the ATP synthase reaction itself is a direct substrate phosphorylation. Now, the electron transfer chain, as we said, consists of molecules that are considered both proton pumpers and electron carriers, and they are present on the cristae of the inner membrane. And that's why we have folds on the inner membrane in order to increase the surface area and allow for the formation of or the presence of multiple molecules of electron transfer chain. I'll make sure we are recording. Okay. Now, as we said, the electrons that are received by the electron transfer chain come from NAD and FAD through a series of redox reactions where we couple oxidation with reduction. And of course, the substance that gets reduced is the substance that attracted electrons, and therefore it has a greater electronegativity. I think we have covered all this. This is important to know. Number 8, no, along the electron transfer chain, every molecule of NAD is or helps or share in the formation of 3 ATP molecules, whereas every molecule of reduced FAD gives rise only to 2 ATP molecules. And this is, of course, there is greater energy delivered by reduced NAD, which means that the electrons in reduced NAD are present at a higher energy level than the electrons of reduced NAD. We have seen a small molecule along the electron transport chain that is called cytochrome C. Cytochrome C is very important. It's present in all aerobes, and this indicates an evolutionary relationship with or a common, as they say in evolution, a common ancestry of those aerobic organisms. Similarly, these enzyme proteins that are called cytochrome C are similar to hemoglobin in that they have a cofactor that is heme, that is very similar to heme. What is a cofactor? A cofactor is a molecule, a small inorganic molecule that activates the corresponding enzyme. This is different from the coenzyme, like coenzyme Q, which is a large organic molecule that helps activating an enzyme. As we said, coenzyme Q is a lipid electron transport molecule. Most of the members of the electron transport chain are proteins, but coenzyme Q is a lipid, and it has another name that is ubiquinone, and ubiquitous means abundant, and this is because coenzyme Q is present in all cell membranes. It has a nonpolar part, as we said, that tends to stabilize coenzyme Q in the hydrophobic interior of the cell membrane, but it has another quinone part, and that quinone part is the part that can receive the electrons, and so the movement of that polar part, which is quinone, will depend upon its redox status, meaning that it will depend upon whether it has received electrons or has gained electrons. So the reduced form of coenzyme Q, meaning that when it gains electrons, it will become more polar, and it will be, when it becomes more polar, as I told you at the beginning of the presentation, it will become attracted to the phosphate heads, and or it will move more towards the phosphate head. What helps this is the fluidity of the cell membrane. So this lateral, and we call it lateral diffusion of coenzyme Q, which is the movement of coenzyme Q from side to side along the cell membrane, as well as its movement upwards and downwards, is helped by the fluidity of the cell membrane. We've talked about coupling exergonic and endergonic reactions. The flow of electrons is exergonic, whereas the pumping of protons is endergonic. We've also talked about oxidative phosphorylation, where we use the energy released from the oxidation of NAD and FAD in order to add a phosphate group at the end of the day to ADP to form ATP through the ATP synthase. So the presence of the ATP synthase is essential for storage of the energy and for coupling of oxidation with phosphorylation. Here there is an important question, which is, what happens if we decouple oxidation and phosphorylation? Sometimes you add a chemical, and this is a famous question, whether in the MCQ or in the free response question. Sometimes you add a chemical, and that chemical decouples oxidation and phosphorylation, meaning that, for example, you might add a chemical that blocks ATP synthase, and therefore oxidation proceeds, but phosphorylation does not proceed. If they both occur hand in hand, then there is coupling of oxidation with phosphorylation, or actually not hand in hand, but if oxidation occurs and the energy is utilized for phosphorylation of ADP, then you have coupled. But if you prevent the final stage, which is phosphorylation, using a chemical that blocks ATP synthase, then you have decoupled the two. If you decouple oxidation and phosphorylation, then oxidation will proceed, but phosphorylation will not proceed. So what would happen? Energy will be dissipated as heat. Energy will, this is a very important question, energy will be dissipated as heat. And another consequence is that ATP will not form. You will not have stored energy in the form of ATP. Is that process beneficial or harmful? Both. In some organisms, that energy that is dissipated as heat is important. In some organisms, I mean, it is important for homeostasis, to regulate the temperature, to regulate body temperature. So sometimes decoupling of oxidation with phosphorylation occurs in living organisms on purpose in order to generate heat, and when it generates heat, it will keep you or keep the organism warm. But if this exceeds a certain level, then this means that all the energy would be dissipated. This first would raise the temperature of that living organism and might harm the cells, and the cells would stop working because of the elevated temperature. And on the other hand, you wouldn't have any stores of energy in your body. So again, decoupling can be beneficial to generate some heat to warm your body, but if it exceeds a certain limit, it would be harmful. And we are going to write this because it is important. We are going to erase all the, so here, decoupling of oxidation and phosphorylation might be beneficial and might be harm. Decoupling of oxidation with phosphorylation might be beneficial and might be harmful. When is it beneficial? To warm the body and therefore it might be important for regulation of body temperature and for homeostasis, but it might be harmful in two ways. It would result in depletion of your ATP stores. It might result in depletion of your ATP stores, and at the same time, it might result in excessive heat, and that excessive heat that is dissipated will result in an elevated temperature, and that elevated temperature would be harmful to the living organism, okay? So the ATP synthase is important. Why is it important? Because protons cannot diffuse. And why can't they diffuse? Because they are charged. They can only flow down the gradient, meaning that the process of chemiosmosis is a passive process of chemiosmosis through the ATP synthase channel. They can flow down the gradient through ATP synthase in a process called chemiosmosis, and this will result in the direct phosphorylation of ADP into ATP. And here we are repeating what I said on the screen when we talked about the three couplings that occur, coupling oxidation with reduction, exergonic with endergonic, and finally here we have coupled oxidation and phosphorylation. And we talked about the concentration gradient, the proton gradient. Proton gradient is an important term that you will be using frequently. Proton gradient, okay? Of course, this is an example of how internal membranes are important for the cell. We talked about oxygen and how oxygen is a final product. And here, if you have the two protons plus the two electrons, after losing their energy, they can now couple with oxygen, with one oxygen atom, to form a water molecule. So this is how oxygen acts as a final electron acceptor and forms a water molecule. Of course, if you multiply by 2, then you will have 4 hydrogen ions plus 4 electrons. Why are we multiplying by 2? Because we need oxygen atoms, 2 oxygen atoms, or actually an oxygen molecule to form 2 molecules of water, okay? So this is how this is why oxygen is important. This is the function of oxygen. It acts as the final electron acceptor, and it helps in the formation of water, and in this reaction, it gets reduced. So again, to summarize, summarize, okay? So here we have oxygen, okay? Oxygen is the final electron acceptor. It is the final electron acceptor in the electron transfer chain. Oxygen gets reduced at the end of the electron transport chain, and oxygen reacts with hydrogen to form water. So the water that is formed is nothing but reduced oxygen, okay? And now we're going to talk, as we said before. Now, of course, this is the overall reaction. It's an interesting diagram. You can inspect it on your own, and it summarizes all the process, and we did the calculations in the classroom last time. We said that we obtain 8 ATPs from glycolysis, and that we obtain 24 ATPs from Krebs cycle, and that the total here is 32, and we obtain 6 from, sorry, here we have, let me repeat it again. Let me do the calculations on a separate screen. So here the total energy yield, the total energy yield of aerobic respiration, or aerobic breakdown of one molecule of glucose. We start by one glucose molecule. So you will have glycolysis, and then after glycolysis, you will have the pyruvate reaction, and then you will have Krebs cycle, and then finally, you will have the electron transfer chain. So in glycolysis, we had two ATP molecules that were obtained directly. This was the net yield of glycolysis, and then by direct substrate phosphorylation, and then we had 2 molecules of reduced NAD as a final product of glycolysis that gave 6 ATPs indirectly. And then in the pyruvate reaction, from the 2 molecules of pyruvic acid, we also obtained 2 reduced NAD molecules, and the 2 reduced NAD molecules along the electron transfer chain gave 6 ATPs. And then in Krebs cycle, we had in the two turns of Krebs cycle, we had 2 ATP molecules directly, and then we had 11 per turn indirectly, meaning that we had, how did we calculate them? We had, per 2 turns, we had 6 reduced NAD, and we had 2 reduced FAD, okay? So the 6 reduced NAD gave us 18 ATP molecules indirectly, and the 2 reduced FAD 2 by 2, this is 4. Let's calculate the total. So here we have 2 plus 6, this is 8 plus 6, this is 14, and then 16, and then 16 plus 18, 34, and then 38. Here, why didn't we put any calculations? Just because all the indirect ATPs, all the indirectly obtained ATPs through oxidative phosphorylation that starts by reduced NAD and FAD and ends by ATP formation along the ETC is the cause of this release of energy, meaning that all the indirect ATPs that have been obtained via the ETC, so we are not going to add any ATP molecules. Why is this inclined like this? Because it shows you how the electrons are being received at gradually decreased energy levels. And then we talked at the beginning about anaerobic respiration, and we said that so here, whenever we want to break down glucose, then we have 2 ways of breaking down glucose. We have an aerobic mechanism of breaking down glucose and an anaerobic mechanism of breaking down glucose. So here we have anaerobic breakdown of glucose to get energy, and we have aerobic breakdown of glucose to get energy. In aerobic breakdown, we started by glycolysis, okay? So here we started by glycolysis, and then glycolysis was followed by Krebs cycle, and Krebs cycle was followed by the electron transfer chain. In Krebs cycle, what happened to the pyruvate of glycolysis is oxidation. So glycolysis led to Krebs cycle, and the major event here in Krebs cycle was actually complete oxidation of pyruvic acid, complete oxidation of pyruvic acid, which resulted from glycolysis. In anaerobic, we also start by glycolysis. But after glycolysis, we don't carry out the chemical reactions of Krebs cycle, but what we do is pyruvate reduction, and there is no oxidation at all, and pyruvate, there is no entry in the mitochondrion, and we stay in the cytosol. So there are major differences here. Of course, anaerobic, there is no oxygen, and aerobic, there is oxygen. This is the first difference. The second difference is that, or actually there is a similarity, they both start by glycolysis. The second difference is that in anaerobic, breakdown of glucose, after glycolysis, pyruvate undergoes reduction, whereas in aerobic, pyruvate undergoes oxidation. So how would we represent this? We would represent this by saying that here we have a process of fermentation or anaerobic respiration. This is important to know. When we say fermentation, we mean anaerobic respiration. We mean anaerobic respiration. But when we say cellular respiration, we mean aerobic respiration. So actually fermentation or respiration or breakdown of glucose that occurs under anaerobic, you'd better say anaerobic respiration, because some references refer to glycolysis as breakdown of glucose or glucose breakdown, which is a very weird term. So actually fermentation is anaerobic respiration. What does it consist of? It consists of glycolysis followed by pyruvate reduction. Glycolysis followed by pyruvate reduction. But if pyruvate reduction, if we reduce pyruvate into lactic acid, then the whole process is called acidic fermentation. And there is no carbon dioxide here. If you reduce pyruvate into ethyl alcohol, here ethyl alcohol is C2, pyruvate is C3, lactic acid is C3. So here there is no loss of carbon dioxide, but here, because there is one carbon less, so there is carbon dioxide that is released, and this process is called alcoholic fermentation. So in acidic fermentation, you reduce pyruvate from glycolysis into lactic acid only, and in alcoholic fermentation, you reduce pyruvate into ethyl alcohol and carbon dioxide. Alcoholic fermentation occurs in yeast, as we said in the early sessions, and carbon dioxide is important to raise the dough because of the bubbles that are produced when you put the yeast in the oven at a suitable temperature, whereas acidic fermentation occurs in bacteria. It occurs in lactobacilli, and we can use lactobacilli in yogurt preparation because of the acidity will help curdling of milk and production of yogurt. But lactobacilli are also present in the mouth cavity. They are friendly flora usually, but sometimes they can cause diseases like, for example, dental caries, when the acidity, when the lactic acid produced will dissolve the enamel in the teeth. And they're also present in the stomach and can be converted into pathogenic bacteria, but rarely. Usually they can live in the stomach because they can withstand the acidic pH of the stomach. In human beings, production of lactic acid occurs in muscles. Muscles, upon exertion, break down ATP molecules to release energy. And when they need more energy, they start to they start to break down glucose anaerobically, and they will produce lactic acid. And that lactic acid will stimulate the nerves, and it is responsible for the muscle pain and the cramps that you feel when you exercise heavily. Why does the muscle refer to anaerobic respiration when you exercise heavily? When you exercise heavily, the heart increases the blood flow to the muscles. The lungs increase their breathing movements. You breathe faster, you breathe more deeply to get in more oxygen, but yet the oxygen and glucose supplies to the muscle are inefficient or inadequate, actually. So the muscle starts to break down glucose anaerobically, which causes the pain and which causes the person who is practicing this kind of exercise to stop in order to wash away lactic acid and diminish the pain. Actually, lactic acid is washed away by the venous flow, and the blood returns to the heart carrying lactic acid. Okay, so these are the only tissues in the human body that carry out acidic fermentation and produce lactic acid. They are the muscles when you exercise heavily. Of course, anaerobic respiration occurred millions of years ago, or started millions of years ago, when there wasn't enough oxygen, or there wasn't any oxygen at all on the Earth's atmosphere. The last thing to say, or the last two important points to say, is that, of course, anaerobic respiration is not effective in producing energy, because every glucose molecule produces only 2 ATP molecules under anaerobic conditions, in contradistinction to aerobic processes that produce 38 ATPs. The second thing is that this process of anaerobic respiration is important to regenerate NAD, meaning that when you produce here in alcoholic fermentation, for example, when you produce pyruvic acid, you have to what is being produced when glucose gives pyruvic acid through the process of glycolysis, you produce reduced NAD. So reduction of pyruvic acid into acetaldehyde and then into ethanol is important, because when you continue and reduce pyruvic acid into ethanol, you are actually oxidizing NAD back into the oxidized form of NAD. So the importance of pyruvate reduction, whether you are talking about acidic fermentation or alcoholic fermentation, is unloading of reduced NAD. So let me erase this because it's a bit messy. So here we have reduction of pyruvic acid into ethyl alcohol. This reduction is important. Why? Because it unloads reduced NAD from its hydrogen, and therefore this unloading is important for regeneration of NAD. Why do we want to regenerate the oxidized form of NAD? In order for glycolysis to proceed, because if oxidized NAD is not restored, then glycolysis cannot proceed, because it can proceed only if there is NAD to carry the hydrogen that is released from glucose, okay? This is the process of acidic fermentation, and it is clear here that pyruvate reduction regenerates NAD. And the last thing to talk about is the oxygen debt. When you respire anaerobically, when you exercise heavily and you respire anaerobically, you produce lactic acid, and that lactic acid continues to build up in the blood. And this lactic acid not only causes pain, but because it causes a kind of acidosis, or acidic pH of the blood, then it will stimulate the respiratory system. It stimulates breathing, and when you stimulate breathing, you wash away carbon dioxide, and when you wash away carbon dioxide, then you diminish acidosis, okay, and you raise the pH of the blood to normal again. And this is what we call the oxygen debt. When you breathe rapidly, you are taking in oxygen. So when you stimulate breathing, first of all, you wash away carbon dioxide to correct the pH, but also you restore oxygen to normal levels, or you restore oxygen to oxidize the lactic acid that has been formed. So actually, you are repaying the oxygen debt. It is as if you had an oxygen debt while respiring anaerobically, and you are now repaying the oxygen debt. The last thing to do is, how is lactic acid removed? How is the lactic acid removed after or washed away? No, removed after heavy exercise, after heavy exercise. So what you do is to hyperventilate, to breathe rapidly. And when you hyperventilate, then what you do actually is to wash away carbon dioxide, as we said before, wash away carbon dioxide, but you can also get in more oxygen, and pay for the debt, and pay for the oxygen debt. How are you going to pay for this oxygen debt? You are going to pay for this oxygen debt by oxidizing the accumulated lactic acid into pyruvate again. And what are we going to do with that pyruvic acid? Use it in Krebs cycle, as there is availability of oxygen now. The second thing is that your heart increases in rate, and the circulation and the blood flow to the tissues increases, and therefore you can wash away lactic acid. You can wash away lactic acid. That was the last item to discuss in cellular respiration. By this, we would have finished cellular respiration. I'm sorry, it was a bit long, but I had to finish the whole stuff in one session. Thank you guys, and see you, inshallah, during the next session. Bye.